Good morning, everybody. Thank you for being here today and for joining us for this exciting science morning. So I'll go over some basics and NARS1 basics, just so hopefully you can understand the scientific talks that are going to be coming after me. Just to reintroduce myself quickly, I'm Ingrid. I'm the scientific advisor for the Rory Bell Foundation. And I did my whole PhD thesis on NARS1 that I finished 2 months ago. And I was in California for all this PhD, and now I'm back home where I'm based in Paris. So if you have any NARS1 science questions, please don't hesitate to ask, or if you have any questions about the presentation. All right, so going very much back to basics, our bodies are comprised of cells, and each cell, in order to divide and to have all the information needed to make brand new cells that contain all the information, have a template recipe, which is called the DNA. And DNA is just like a cookbook. So the cookbook contains all the information to remake every cell in the body. But when you're making different tools that are going into cells, you do not want to touch the original cookbook so it doesn't get altered to pass on to different cells. So that's why we have RNA, or just meaning it is taking a little chunk of DNA or a gene and taking this information, and you can see it as a recipe for a cupcake, for example. And I'll keep this analogy so you can understand, hopefully, what I've been talking about. So the RNA is the information on how the final functional tool is going to be made. So this is how the recipe that has the different instructions for what's going to be made. And then the final tool that is going to be the functional tool that's going to be helping in the cells, or like giving all the information, is the protein. So here, the cupcake, or the final product of the recipe, if you will. And then if you put all those desserts, all those recipes together, and then put them into a global thing, then you have tissues, or like, for example, liver, or brain, or different organs. So where does NARS1 sit in all this? So NARS1 is the acronym for asparagine tRNA synthetase. And NARS1 is one of the little piece of the gene, or DNA, that is in the original cookbook. And we each have 2 copies of this original DNA. Now, NARS1 is the gene, and then the final product of NARS1 is the protein, and you'll see it as different names, so sometimes it can get a little confusing, but NARS1 is the gene name, and then AsNARS, or asparagine tRNA synthetase, is the protein name. Just for the sake of simplicity, I will refer to everything as NARS1, just so it's easier for everybody. But yeah, final product is the protein, so NARS1 is a protein. And is a tool that is functional. Right. So each protein, including NARS1, has a definite role in the body, allowing the body to grow and to function. Some proteins are non-essential for life and are more used, for example, to differentiate like a liver cell or brain cell that have different needs, where NARS1 is very crucial and very important for every single cell in the body. If there's no NARS1, there is no life possible. And just to complicate things a little bit further, NARS1 is actually a protein that helps make proteins. So it acts here in between the RNA and the protein. So if you don't have any NARS1, or AsNARS, you won't have any protein, so you won't have any functional tools in the cell, and the cell is not going to be able to function.
So basically, a bowl of batter.
Basically, or just, you don't even go past the recipe stage, or just ingredients, yeah. Just to get a little more sciencey, and I promise that's the most sciencey I'll get. This is what NARS1 looks like, here in blue. So this is what it actually looks in the cell. And NARS1, so I just mentioned, contributes to protein making and has a specific things that works together with NARS1, which is asparagine. And you'll hear more about asparagine in the next talks. So asparagine here in purple and tRNA here in green are being put together by NARS1 right here. And then these two are going to be able to make proteins, like right here. And then NARS1 gets recycled, and so you can put more asparagine and tRNA together. Right. Now, what is asparagine? So proteins, like NARS1, are made of single individual building blocks that are called amino acid. And asparagine is one of those 20 amino acids. So like, for example, here you have a protein being made, and you have asparagine right here, and then you have other amino acids, which determines the identity of the protein. So like, different letters or different amino acids are going to make different proteins. And it is put together in a very precise order, where every single letter matters. You'll see asparagine right here. Asparagine, just as NARS1 has multiple names to make things easier. So asparagine, ASN, or N, and you'll be hearing more about it because it's really specific to NARS1. And every single protein in the body pretty much contains asparagine, which is why it's important to have NARS1. And if you change even one amino acid in the recipe, then you can change the entire recipe. And I'll go over that. Right, so here you have a protein, and then little single building blocks are the amino acids. Now, what are variants, or when you change one amino acid to another? So when the recipe changes, the final product can be affected. And I'll have some example. And it usually happens at the recipe book, so at the DNA level. So every single NARS1 in the cell that has been changed is going to contain the same variant as the same information. So yeah, if you change even just one little letter, you can change the entire process in the final recipe, and I'll go over that. So bear with me, I'll explain the table. I'm sure you all have heard of different variants that you can, within the NARS1 disorders. I'll take the example of the title of the recipe, which is the carrot cake muffins. So you can have a silent variant. Here you can change the letter, so a C to a K, and it's going to go and make carrot cake muffins. So spelling is different, meaning is the same. That is a case where the proteins out of the NARS1 that is made is the same one. And in this case, keep moving. But in this case, it doesn't have any impact. Well, you have missense variants. So for example, a C gets changed to a P here in the carrot, and you change from carrot cake muffins to parrot cake muffins, which is very different. So you still have a recipe made, you still have something being made, but instead of carrot muffins, you have parrot muffins, which is a different information, a different protein. Now, a little more complicated, which is the insertion and deletion. So you add letters within the title of the recipe. So for example, you can add 4 letters between the 2 R's of the carrot, and then you'll have something that doesn't mean anything. And in this case, usually the cells recognizes it and doesn't make any NARS1 or no protein. There's another case where, for example, if you change 3 very specific letters and that still have a meaning, for example, if you change the RRO, again, you have a different muffin made, which is a cat cake muffin. Still protein, but very different. And then the nonsense, which is the case of the RR534, for example. There's 2 cases. Either you have a stop, so a shortened protein. For example, A2 stop, you just end up with a P, so you have no protein at all. But in another case, you can have a stop that is at the very end. So you still have most of the information. Right here, you have your carrot cake muffin. So you can still guess what's going on, but the protein is functionally going to be very altered. So it can have small differences, can have very large impacts, and is happening at the NARS1 level. Right. Now, inherited, yes. Is there a question over there? Yeah. Regarding that?
She's just going to identify.
No.
What kind of muffin she is.
Is this one working? Oh, this one's working. Okay. No, I was just going to say, I think amongst our community, we're always identifying the kids and saying, you know, what does my child have versus someone posts about their child, and you can see different abilities of the kids based on where their mutation is on the gene, what's being changed. So I know what Grady is, but a million times Rachel has to remind me, like, Grady is a missense. He has 2 bad copies of the gene. So basically, different proteins being made, 2 missenses. He's more affected than a lot of our Vincenzos, Eleonoras, Kians of the world who just have a nonsense where there's just one stop at the end. And so I think depending on, you can get a better understanding here of how much different from the recipe your child is based on what they have, what their mutation is, if that makes sense. It's just my non-scientific translation of what you shared is, I think, how we all within the group, as we meet new people and hear, you know, what variant or what mutation they have and are trying to process, you know, what that means, whether they have 1 or 2, or how off maybe their recipe is from the normal original mixture.
You can just add that NARS1 is unique, actually even in the Rs genes too, because we have, you know, groups that you know, the kids just have 1 gene that's affected, right? So just a carrot cake muffy only, and we also have a group where us as parents are carrying a variant and we're passing it down. So Terry's a parrot, and Chris is a parrot, and now Grady now has a beak and some talons in his muffin. Yeah, so that's kind of that, and I think, again, just to frame it from a Rory perspective, which is even different, I'm actually a silent, so technically it's a carrot with a K, but she got a carrot with a K, which is really bad, because now she's got nothing, and then her dad was a parrot, so she had just a tiny little bit that was made. So if that helps, nobody wants to eat beaks in their muffins.
Pretty much.
I don't think, yeah.
Makes sense.
Yeah.
Okay.
Thank
Sorry
you.
to interrupt.
No, I'm glad you understood it. All right. Yeah, so now moving on to inheritance and what Terry and Rachel was just talking about. Thank you. Little introduction. So as I mentioned before, within the cookbook, like the original DNA, we have 2 NARS gene copies here in blue that can contain variants that I call it here in orange. There's 3 different cases that we see within the NARS1 disorder inheritance. So first one, where both unaffected parents carry a variant here in orange that most of the time they're not aware of, and then passed on to their child those 2 variants right here, where the child is affected by NARS1 disorder. What we call that is, so the parents are heterozygous, meaning they have 1 blue and 1 orange. So like 2 different copies of the NARS1. The child is homozygous because it has the 2 same variants. And we call this variant recessive because you need 2 of the variants in order to see a NARS1 disease phenotype. And in this case, it's inherited because it was passed down from the parents. So one of the variant that we see that is transmitted like that is, for example, the R545C variant. Second case where both unaffected parents are passing down a variant here in orange and a different variant here in red. So it's not passing down the exact same variant, but the child is affected because both variants are not functioning properly. So in this case, the parents are also heterozygous. That means they're carrying one blue, one orange, and one blue, one red, so like two different ones. And the child is not homozygous, but compound heterozygous, meaning that the two variants are different, but together cause a NARS1 disorder. And this is also recessive because you need two variants in order to see a phenotype. And this is also inherited from the parents. And the last case, which is when unaffected parents are carrying two healthy copies of the NARS1 gene, like here in blue, and somewhere in the development of the child or the embryo, there is a new variant that appears and that is not passed down. So it's de novo, it's not inherited. And in this case, the child is heterozygous, so he has one healthy NARS1 and one variant right here, de novo, and the parents are homozygous because they have the same copies and the child is heterozygous. Hope that makes sense. Right. So just switching gears a little bit. Now, why do we need research and what we've been doing over the past years? So of course the goal is to find treatments. So there's strategies that have been worked on, so like drug repurposing, ASOs, and we'll be hearing more about this later, asparagine supplementation. But why is it important that we have research and we have researchers working in the background? Well, first, if we want to find a treatment, first we need to figure out outcome measures. We need to figure out what is the most important thing that you want in your child, that you want in patients, like what is the most important symptoms that you want to see treated. And for those, it's important to, of course, fill out the surveys, but also if we can make models, for example, mouse models or other models other than patients to figure out the different symptoms. We can identify outcome measures and we can identify biomarkers that are going to allow us to test drugs and to see exactly what we want to go down or what we want to go up, right? What already approved drugs could work? So we have this thing called drug repurposing, where drugs that are already on the market could be working on the symptoms of the different patients. But how does one test if all of these drugs that are approved could work on the symptoms? Obviously, you don't want to test like 1,000 drugs on your kid and just figure out what's going on, right? So that's why we need models. We can do that in mice, so we can have a mouse that is carrying the variant, and then we can test those drugs and see what's going on, right? That's also why we have cells, like the cells derived from patients, where we can test those drugs and see if there's an effect without harming patients. And there's also other models like fibroblasts from patients and zebrafish. All those contribute to having more models that are not harmful to patients to test those different things. What is the best dose for treatment? Let's say we find something. How do we determine what's toxic and what's working, right? We don't want to right away just give a drug to somebody and figure out there's toxicity after, right? So also, that's also why we're trying to have models in the back, and we're trying to have, again, the same models, and that helps us also to set the goal for clinical trials. So this is called preclinical trials, so we can set the doses and be like, okay, here is how much we can give to one organisms and figure out the safe dose so we don't include toxicity on top of the treatment, right? And then another one is, can variant severity be predicted? Like, you just got a NARS1 diagnosis, you just got a new variant. We don't necessarily know what it means. And so that's why we in the lab can make proteins, so we can make recombinant proteins and make NARS1 in test tubes to figure out what's going on at the activity level. We have some researchers doing some functional experiments to try to figure out, does it work with asparagine, with tRNA, and develop different assays. So that's what researchers are doing in the background, and they'll be talking more about this. Now, where are we at with all this? So this is the research roadmap for the Rory Bell Foundation. So everything that Rachel and Terry and everybody has been working really hard on. I'll be talking about the research and development here. Oh, sorry, that's this plasma right here. But here you have the little boxes ticked of all the things that are in the pipeline. So they've been working hard on. So we have functional experiments, we have assays, and then our researchers are going to be talking more about this. We're working on drug repurposing. So this is what I just mentioned, which is the drugs that are already approved on the market, but we see if maybe one of them is working on somebody's symptoms. And then curative therapies. And then we have been working also on the animal and cell models. So like the different models to be able to test all the things and all the hypotheses we've been thinking about. We're actively working on making mouse models for 2 of the variants. Cells, which are cells derived from patients, so iPSCs derived from patients that are allowing to, for example, recreate neurons or recreate other types of cells so we can test specifically what's going on into different organs. Same thing for fibroblasts, and then plasma levels, they're allowing us to test for NARS1 activity. And then also, we have biomarkers and so on that we'll be hearing more from the researchers. And that's it for me. Please let me know if you have any questions. Be happy to answer. Thanks. Thanks.